In the last few lectures, we covered cell-mediated immunity, which involves T lymphocytes. Cell-mediated immunity fights against intracellular antigens. They kill infected cells, tumor cells, etc. But there are many pathogens, which multiply in the extracellular spaces of the human body. These extracellular spaces are protected by B lymphocytes, which are responsible for humoral immunity. In this video lecture, we will understand role of B lymphocytes and the basic structure of antibody. B lymphocytes originate in bone marrow, and they also complete their maturation in the bone marrow. After the maturation stage, these cells are released into the blood. They keep recirculating between lymph, blood, and secondary lymphoid tissues. When these mature B cells recognize specific antigens, they get activated. This recognition occurs via specific receptors present on B cells, which are known as B cell receptors. Once activated, these B cells proliferate and differentiate into effector B cells and memory B cells. Effector B cells are known as plasma cells, which are antibody-secreting cells. They produce and secrete antibodies specific to the antigen recognized by the B cell. Antibodies are glycoproteins. The basic structure of antibody resembles Y-shaped molecule having two antigen binding sites and a stalk known as Fc region. These antibodies circulate in the lymph and blood. From there, they reach the site of invasion by the pathogen. They bind to the pathogens or antigens and activate defense mechanisms that lead to the destruction of the pathogen. Antibodies are also known as immunoglobulins. This is because they belong to a group of glycoproteins known as globulins. The term immune reflects that these glycoproteins play a major role in immunity. The basic structure of all antibodies and the structure of B cell receptors is same. So, B cell receptors are also immunoglobulins. The difference is that B cell receptor is a membrane-bound immunoglobulin, whereas antibodies are secreted immunoglobulins. Let us now study the basic antibody structure. All antibodies have a same core structure. It consists of 4 polypeptide chains. 2 identical heavy chains designated as H chains, and 2 identical light chains designated as L chains. As you can see here, heavy chains are the longer ones and light chains are the shorter ones. The term heavy and light refers to their molecular weights. Heavy chains have more molecular weight than light chains. Now, since these are polypeptides, the N-terminal of this polypeptide chain is present at the tip and C-terminal is present at the base of each polypeptide chain. As you can see in this image, these chains are assembled into a Y-shaped structure. Each light chain is connected to a heavy chain via a disulfide bond. Here, this yellow line is representing a disulfide bond. The heavy chains are connected to each other via 2 disulfide bonds in the mid-region. Besides these disulfide bonds, a number of non-covalent bonds are also present which keep these chains together. The mid-region of antibody has considerable flexibility. This region is known as the hinge region. The main advantage which this region provides to an antibody molecule is the ability to adjust to different spatial arrangements of antigens or epitopes. In other words, hinge region make possible the rotation and bending of antibody molecule. The stalk of this Y-shaped antibody molecule is the stem region, also known as Fc region. To each heavy chain, short carbohydrate chains are attached. These carbohydrate chains serve many additional functions such as increasing the solubility of immunoglobulins. Before going into further details of antibody structure, let's have a look what we have studied just now. These are the two identical heavy chains and these are two identical light chains. Each light chain is attached to a heavy chain via a disulfide bond. The two heavy chains are linked to each other via two disulfide bonds. Mid-region is known as hinge region and this is the stem or Fc region. Short carbohydrate chains are attached to each heavy chain. Here you can see how 3D structure of antibody looks like. Let's study more about light and heavy chains. Light chains. As we said before, these are the two shorter subunits of basic antibody molecule. Each light chain has a molecular weight of about 25 kilodaltons. And each contains about 220 amino acids. In humans, there are two types of light chains: kappa chain and lambda chain. They are similar in structure and function, but they are coded by different genes. Kappa chains are encoded on chromosome 2 and lambda chains are encoded on chromosome 22. It is important to note here that each antibody molecule produced by a B cell will either have kappa or lambda light chain, but never both. In humans 60% of light chains are kappa and 40% are lambda. Heavy chains. Heavy chains are the longer subunits of the antibody structure. Each heavy chain has a molecular weight of about 50 to 70 kilodaltons. And each heavy chain contains about 440 amino acids. There are 5 types or classes of heavy chains in humans, all encoded on chromosome 14. These 5 classes are designated by lowercase Greek letters, gamma, alpha, mu, delta, and epsilon. They are also written as G, A, M, D, and E, respectively. Each light and heavy chain contain 2 distinct regions: variable regions and constant regions. Variable region refers to the first 110 amino acids of the N-terminal region, in each heavy and light chain. These regions are so called because the amino acid sequences in these regions have great variability. These regions are designated as VL in each light chain and VH in each heavy chain. It is the variable region of a light chain and a heavy chain, which together form the antigen binding site. So, there are 2 antigen binding sites in a core antibody molecule. Further, the variability of amino acid sequences in these regions is localized within certain areas. Since these areas consists of variable amino acid sequences, these areas are known as hypervariable regions. They are also known as complementarity determining regions, abbreviated as CDRs. This is because these regions together form a structure which is complementary to the shape of specific antigen bound by the antibody. Let's say this line is representing first 110 amino acids in the variable region. The CDRs are represented by blue color. Scientists have found that there are 3 CDRs in the variable region of each chain. These are designated as CDR1, CDR2, and CDR3. The intervening sequences between CDRs or hypervariable regions are known as framework residues. These intervening sequences have restricted variability. Now, as you can see here, the CDRs are the regions which actually interact with its specific antigen, or in other words, they form the antigen binding site. These CDRs are brought together when the antibody molecule folds into its native conformation. In this image, the three blue color lines in each light and heavy chain is representing the CDRs. So, now we understand that antibodies can recognize diverse types of antigens because of these hypervariable regions. The region beyond the variable region of both heavy and light chain is known as constant region. They are so called because the amino acid sequence in these regions shows little variation among antibodies. There is a single constant region in each light chain which is designated as CL. Now, recall that light chains are of two types, kappa chain and lambda chain. These chains differ from each other by minor differences in the constant region of light chain. Constant regions in heavy chains vary from 3 to 4. This depends on the antibody class, and it is the constant region of heavy chain which forms the basis of this antibody classification. In a particular class of antibody, all antibodies have almost same constant region, but constant region of one antibody class is different from the another class. The constant regions of the heavy chains are designated as CH1, CH2, CH3, CH4 starting from the end terminal of the chain. Finally, each variable and constant region in an antibody molecule also has at least one disulfide bond. These are internal disulfide bonds. You must have come across the terms Fab and FC in immunology. What are these? To understand these terms, we need to look at key experiments which revealed the antibody structure. These experiments were: proteolytic treatment of immunoglobulin or antibody with enzymes papain and pepsin. Chemical treatment of immunoglobulin with the chemical mercaptoethanol. Gerald M. Edelman and Rodney R. Porter shared 1972 Nobel Prize in Physiology or Medicine for determining chemical structure of antibody. Let's understand these experiments and their results in brief. First, we will see what happens when an immunoglobulin is treated with the enzyme papain. Porter did his experiment on immunoglobulin G of the rabbit. Papain is an proteolytic enzyme. As the name indicates, it splits or cleaves proteins, and we know antibodies or immunoglobulins are also proteins. When the antibody is treated with enzyme papain, the antibody splits into 3 fragments of about equal size. It was found that enzyme papain cuts the antibody molecule just before the two disulfide bonds, which link the heavy chains together. That is, at the amino terminal side of interchain disulfide bond that link two heavy chains. As a result, two identical fragments are produced. Each of these consist of one light chain, and one heavy chain held together by interchain disulfide bond. Since these fragments have antigen binding activity, Porter called them fragment antigen binding, abbreviated as Fab. So, there are two Fab fragments. The Fab fragment contains the V regions and binds antigen. The third fragment has no antigen binding activity at all. It consists of constant regions of heavy chains, held together by disulfide bonds. It was found that this fragment crystallized during cold storage. So, it was called as fragment crystallizable, abbreviated as FC. We now know that FC fragment play role in opsonization and complement fixation, which involves antibodies. Edelman treated immunoglobulin with the reducing agents such as mercaptoethanol, and he found that antibodies contain disulfide bonds. Mercaptoethanol is a reducing agent and it breaks disulfide bonds. As a result, all disulfide bonds are broken, leading to the splitting of immunoglobulin into two identical chains with a molecular weight of about 22 kilodaltons each, and other two identical chains with molecular weight of about 53 kilodaltons each. The larger molecules were designated as H or heavy chains, and the smaller ones L or light chains. In other experiment, Albert Nisnoff treated immunoglobulin with enzyme pepsin. It is also a proteolytic enzyme, but it cleaves immunoglobulin molecule at the carboxy terminal side of the interchain disulfide bonds that link heavy chains. This results in a fragment, in which the two antigen binding arms of the antibody remain linked. This fragment is represented as FAB prime whole twice. Now, here you know F stands for fragment and AB stands for antigen binding. The prime symbol here represents variation in the structure of this fragment as it contains a few more amino acids than the Fab fragment. The number 2 here represents that two such fragments are linked to each other. This fragment has same antigen binding ability as the original antibody molecule. The remaining part of antibody is cleaved by pepsin into several small fragments. So, now we understand what are Fab and FC in an antibody molecule. There are two Fab in basic antibody structure. These are antigen binding fragments which contain variable regions and the first constant region of the N-terminal side. The FC is composed of only constant regions of heavy chains and form the stem of basic antibody molecule. Note that C in FC is representing the term crystallizable, since this fragment crystallizes under cold storage.